Thank you, Shiro. Thanks once again to Shiro and our Japanese hosts for their splendid hospitality on this event. And thanks to the audience for hanging in here for 3 full days. I'd like to talk to you about my perspective on the human connectome and how it relates to the neuroinformatics challenges that we face. We've heard the analogy of cartography. We've had a millennium in which it started out with classical, beautiful but inaccurate maps. I grew up with maps of the world that were embedded in book atlases. The satellite and digital imaging explosion brought us Landsat, and then the spectacular power of Google Earth that let me look at where I started and where I ended on this trip in fast order. We've had a mere century of brain cortical cartography, starting with the classical anatomists, Brodmann and others, a period when we used book atlases of the human brain, and now the explosion caused by the revolution in neuroimaging with structural and functional MRI and other modalities and analysis methods that are taking us into an extraordinary period with great opportunities for deep and powerful data mining. Now, the human brain we've heard about it before, that it has about 86 or 90 billion neurons. We have good numbers on that have been obtained by accurate methods of cell counting, and the portions of the brain are dominated by the cerebral cortex, which contains 80+% of the brain mass, but only a small minority, about 20% of the neurons. It's got some 150 trillion synapses and 160,000 kilometers of axons lurking in there. The cerebellum is tiny, but contains 80% of the neurons, and the rest of the brain contains a mere less than 1% of the neuronal count. Now, I'm going to be talking a lot about the human brain, but I'd like to start with the brain of the macaque monkey. That gives us a lot of baseline information. And I want to talk about parcellations and also about connectivity. And then I'll move up to human cortex, monkey-human comparisons, and then human-brain connectivity. So, the parcellation of the cerebral cortex in monkeys and other species has been done for more than a century by a variety of methods: functional, architectural, architectonic, connectivity, and topography. And within each of these broad approaches, there are multiple modalities that provide critical information. And what we get are parcellations of the type that you see here, of large, medium, and tiny areas by one or another parcellation scheme. And the primary data from which these are derived are things like architectonic analyses of tissue sections that can be reconstructed and mapped extensively onto an individual subject's brain or macaque hemisphere. And then we have to bring the data from multiple sources into a common framework. And one general strategy that benefits from surface representations is that we can respect the topology of the cortical surface and register an individual to an atlas using various methods. In this case, we're using a landmark-driven approach to deform an individual flat map to a target flat map. Nowadays, we mostly go through the intermediate step of projecting data from the surface onto a spherical map and registering one sphere of the individual to the atlas sphere so that we avoid the problems caused by artificial cuts in a map. But the basic point is we have now increasingly accurate methods for registering individuals to atlases, and we can take data then from many subjects, in this case 5 architectonic maps superimposed on one another, to generate probabilistic maps that represent individual variability, and we can extract from that summary representations that show a composite, most likely borders of many different areas by a particular parcellation scheme, and we can use software tools like our Caret software to point and click and see what is this yellow area and what do we know about it and where's the publication from where that came. But we're still in the stage in 2010 of having all too many distinct parcellation schemes that are in widespread use. And so here are 3 of many currently used parcellations of macaque monkey visual cortex, and the regions where they're in good agreement are relatively just a minority of the total number of areas, and in many other regions we have to recognize that we don't know what ground truth is. We can choose amongst various competing parcellation schemes. But even with that limitation in our current understanding, there's a lot we can say about the circuitry revealed by pathway tracing studies. Now, nearly 2 decades ago, when Dan Fellman and I surveyed the literature, we've found this evidence that you've already seen for several dozen parcellations of parcels of visual cortex, and we found that there was evidence in the literature for hundreds of distinct pathways or connections from one area to another. And this is the more commonly visualized version of that hierarchy and connection scheme. But we also published what in retrospect turns out to be the first version of the macaque connectome, a connectivity matrix that showed pluses for every existing connection between one area and another possible target. So that was useful, but it was also limited because we basically had precious little information about the strength of connections, even though we were fully aware at the time that there's a wide range of connection strengths. And so a decade later, Jim Lewis and I published maps that showed more quantitatively the connection density from retrograde labeling studies, and you can see there's weak, medium, and strong connections. There was more quantitative data there, but it was hard to analyze, but we knew that there was orders of magnitude worth of connection strength. More recently now, in 2010, there's an impressive set of analyses by Nikola Markov and many others in Henry Kennedy's lab that have revisited using a more systematic and compulsively thorough analysis of connectivity in macaque monkey revealed by retrograde tracers. And so they've digitally analyzed all of the connections associated in this case with area V1, and they've done this repeatedly in many animals, replicated the analyses, and shown, for example, that area V1 is connected not just to 5 or 10 different areas, but to literally 25 areas consistently across multiple repetitions of the experiment. And what you see here is a logarithmic plot showing that the fraction of labeled neurons ranges from almost 80% for the strongest pathway, V2 to V1, down to 5 orders of magnitude weaker, and yet the error bars show you that these are consistent pathways. So this is telling us something serious and sobering about the diversity and range of connection strengths. And when we look at this similar patterns for 3 different areas, V1, V2, and V4, you can see that each of these areas has a couple of dozen sources of inputs from this type of analysis, and that there's a huge overlap in terms of the commonality of areas providing inputs. but the difference between areas is in the strength of the connections. So each area has a signature or a profile of the type that you see here, and that's the anatomical reality that we're learning from macaque monkeys that we need to carry forward as we contemplate, as I'll get to in a little bit, about the human connectome. This is one more representation of the strength of pathways from one cortical area. For example, V4 is connected to this set of areas in the macaque by this logarithmic scale from showing the strength of connections. So we're able to get a more advanced understanding of the macaque connectome from modern application of retrograde tracer techniques. We have another powerful method that has been validated in monkeys and studied in great detail in humans, as I'll get to a little bit later, and that is resting state fMRI. So it was discovered back in the 90s that if you put a human subject in a scanner and just have them do no task but record the bold imaging signal, that there are slow fluctuations in the signal at any one location or voxel, and many other voxels show a strong correlation in that signal, and other regions, other voxels show no correlation or even an anti-correlation. And when this was done by Vincent et al. In the macaque, a seed location of a set of voxels in the left interparietal cortex showed a region of strong correlation nearby and on the opposite hemisphere and up in the frontal lobes. And that was a consistent pattern, and it could be mapped onto surface representations and shown that this pattern of resting state fMRI correlations is strikingly similar to the task-evoked activations when a different set of monkeys was asked to do an eye movement task, and it's strikingly similar to the anatomical connectivity that I just told you about from Jim Lewis's studies. So in a nutshell, resting state fMRI is telling us information that is related to anatomical connectivity, but it's not just direct pathways, it includes indirect pathways for sure. I'm not going to show you the evidence, just assert it. Okay, let's move up to humans now, and first of all make a few important comments about individual variability. If you take two brains, their MR scans, and put them in the same size shoebox, that is, register them to a common atlas target, you see the detailed pattern of cortical folding is not aligned, and that's because the folding pattern is highly variable from one hemisphere to another and from one individual to another. And in fact, that's just one aspect of variability. Brains in this room, everyone has a brain of a different size and shape, of a very different cortical folding pattern. The individual cortical areas, such as V1, vary dramatically by 2 to 3-fold from one individual to another. There is a great deal of variability in how an area such as V1 in red relates to the folding pattern from one individual to another. And then, of course, we presume that there are tremendously significant differences in the brain circuitry, the wiring that makes each of us unique in terms of our cognitive and other processing capabilities, whether we're good or bad at certain things. And I'll come back to that. But coming back to the folding patterns, if we look at a bunch of individuals and this mildly inflated surface, you see that these folding patterns, even when we smooth them out, there are only a few that are highly consistent, and in most regions there's just tremendous variability that's almost like the cortex was crumpled to get it there. A quick aside about why the cortex folds and what it signifies. Cortical folding occurs in the last trimester of human development, from a lissencephalic to a very highly folded brain at birth. And a hypothesis I published more than a decade ago proposes that this folding occurs because of literal mechanical tension, physical tension generated by axons as they go from their cell bodies to their terminations. And that coordinated axonal pulling, I hypothesize, can bring strongly connected regions together and let weakly connected regions form a sulcus in between them. And this one hypothesis can account for both the consistency and variability in convolution patterns. That consistency occurs in regions dominated by large areas like V1 and V2, and variability may occur in balkanized regions where there are many small areas competing for the strength of their connections to drive certain folding patterns. And lastly, abnormal connectivity or differences in connectivity may lead by this hypothesis to differences in cortical folding. So we can explore those issues if we have atlases for human cortex that cope with this high degree of variability. And my lab has generated a particular population average landmark and surface-based human cortical atlas that you see here. And basically we take a bunch of hemispheres and generate an average surface after registering each of these to a common target. And then we can view all sorts of data on inflated or highly inflated surface maps of the type you see here. So we can take the classical Brodmann scheme and paint it on our PALS atlas. We can take more modern parcellation schemes like Joel Price's orbital frontal areas, register them to the atlas. We can take modern Fmri activation patterns and visuotopic maps, register them to the atlas. We can take disease conditions such as Williams syndromes and look at individuals on their MR volumes or their surface representations and say it's hard to see the difference looking at them one at a time, but we can use surface-based analysis methods to identify folding abnormalities alluded to before and now shown here for Williams syndrome to see several dozen regions where there are specific abnormalities in the Williams versus control populations. We can do that on multiple disease conditions and see that Williams syndrome has a folding abnormality that shows up in a common location with Asperger's syndrome from autism. But other types of autism, low functioning and high functioning autism, have a different pattern of folding abnormalities that are more similar to the folding abnormalities associated with schizophrenia. And so in general, we can see that bringing data onto surface-based atlases provides an incredibly rich framework for exploring folding abnormalities and functional characteristics and many other types of data that are at hand. We can take the human and monkey data that I've shown you sequentially and put them side by side, literally here, and ask how do the maps of cortical parcels in monkey relate to the maps that look very different side by side in humans, and what areas can we say are homologous and what can we say about the evolutionary divergences. To do this, we can identify a set of strong candidate homologs, V1, V2, MT, auditory cortex, and so on, and use those to constrain a mapping from macaque to human, and we can then register the macaque to the human and deform a macaque to a human surface, and notice in the way that some regions seem to expand more than others. We can be quantitative about that and identify regions of hotspots of cortical evolution where human cortex is vastly more expanded by 20 or 30 fold compared to the most likely homologous regions in the macaque. So that's an area for, that I'm quite intrigued by exploring, but I'd like in the last section of this talk to get into the issue of human-brain connectivity using a set of increasingly powerful imaging modalities. One of them relates to diffusion imaging and tracking the white matter bundles that traverse from one gray matter region to another. You can see in this classic partial dissection that there are many places in the white matter where bundles stream along together, but in other regions the fibers are crisscrossing, and in general we have a tremendously complex tangle of pathways to try to elucidate, to understand how this hundreds of thousands of kilometers of wiring is organized in terms of pathways linking perhaps 150 or 200 cortical areas by thousands of cortical pathways. Well, how do we get at this? We now have the methods of diffusion imaging, which can analyze for each voxel of white matter what is the trajectory of the dominant fiber bundle, and in regions where there are crossing fibers, the analysis methods developed by the Oxford group and others allow for each voxel to say, well, the dominant fiber is going this way, but there are other fiber bundles that are going in 2 or 3 other directions. And so for a single voxel, one can connect the lines and identify multiple pathways intersecting at that voxel, but connecting one brain region with another. And so using this kind of strategy, one can identify, for example, a well-known classical fasciculus, the arcuate fasciculus, that connects the temporal and frontal lobes, and we can analyze that with tractography, and then we can combine this volume-based initial analysis with surface maps that show, for example, this little spot right here has an inferred connectivity pattern with nearby temporal and parietal regions and distant frontal regions through the arcuate surface. So we can do that for structural based approaches of diffusion imaging. We can side by side analyze the same subjects for resting state functional connectivity of the type I told you about in the macaque and has been done extensively in humans. And we can approach this now as part of a systematic assault on understanding the human brain circuitry, the human connectome project. And we are very optimistic that NIH will, in the next couple of weeks, announce a major grant to a consortium effort that will chart the human connectome in a large number of normal adults, make the data publicly and quickly available, and face the extraordinary set of challenges to make this data as accurate and analyzable as possible. But if successful, we think it can be transformative for systems neuroscience, and for sure this kind of endeavor demands very sophisticated neuroinformatics, that some of which is still to be invented. So NIH has indicated it is anticipating to award 2 grants, the larger of which we expect to come to the WashU, Minnesota, or WUMIN consortium that altogether has 9 institutions and several dozen investigators, but led by data acquisition at WashU and Camille Ugerbill, the other PI at University of Minnesota. These are the other groups in the effort. The substance of what we're going to tackle is to use diffusion imaging followed by tractography analysis and resting Fmri that I've told you about as the 2 primary methods of learning about the connectivity. But in all of the subjects we will also carry out task-evoked Fmri by a battery of tasks, and in a subset of subjects we'll send them to St. Louis University for magnetoencephalography and electroencephalography analyses. Every subject will be analyzed with behavioral tests using the NIH toolbox supplemented by several other tests. We will gather blood samples and at the end of year 5 at a minimum do genotyping, and based on what Lee Hood was telling us, I'm hopeful we'll be able to convert under budget and do that to full genome sequencing, but that's on our wish list. But this is an endeavor where we intend to study a large number, that is 1,200 healthy adults. By the stipulation of NIH, they want baseline data on the healthy adult condition before heading off in the direction of disease and developmental and various disorders. But coming back to our core mission, we've devised a twin-based strategy to get the maximum bang for the buck. So we will scan 300 twin pairs, and for each pair, 2 of their non-twin siblings, and that will give us maximal power for analyses of heritability of connectivity, and also for genome-wide association studies will benefit from this twin-based strategy. We intend to do this on a multi-platform basis. We have ordered a scanner from Siemens that is customized for improved data acquisition capabilities, and that will end up at WashU and be used to scan all 1,200 subjects. In addition, Ugerbel's group at Minnesota has independently acquired a new 7 Tesla scanner, and we intend to send 200 subjects up for high-resolution 7 Tesla scanning. As I mentioned, some of the subjects, 100 in all, will be scanned with MEG and EEG at St. Louis University. And the kinds of approaches that we want to bring to bear is, for example, to compare resting state fMRI and tractography in detail in every individual subject. So here, for example, for this black node here, this is the pattern of resting state fMRI correlations and anti-correlations from that seed location. And this is the pattern of the diffusion tractography connections in the same subject using what are very preliminary analyses. They are not optimized in the way that we will by the end of phase 1 of our Connectome project, but we will be able to compare connectivity for that location and for every other location, and note, for example, that up on the frontal lobe the pattern of connections is substantially different, but we already have a part of the toolkit we'll want for analyzing and real-time comparing of connectivity patterns. We will also use connectivity data to drive the analysis of cortical parcellation. So the methods I mentioned before for parcellating the cortex are powerful, but they can be enhanced by using differences in connectivity to compare one region's connections with another, and to look for gradients where connectivity patterns change sharply. And once again, showing you preliminary data using this gradient-based approach that's been pioneered by Alex Cohen in Steve Peterson's lab, one can identify, in this case on a flat map of the Atlas cortex, hotspots or streaks where connectivity pattern changes dramatically from this location to that location across this gradient boundary. And when we look for validation by comparing the cortical pattern of activation in a group of subjects doing a saccade task, what you can see at a glance is that the transitions in the connectivity gradient from the functional connectivity analysis tend to coincide with the boundaries of task-based activation. So this gives us encouragement that we're on the right track, that these are real biological signal, not just a lot of noise, and we will be pursuing that as we go forward and use the connectivity patterns to parcellate the cortex of individual subjects, to identify the centers of those parcels, and to carry out network type connectivity analyses of the type that you see here using software analyses that Olaf Sporns and our consortium provided. So this is a structural connectivity matrix between a set of parcels in this individual subject, if we take from the same subject the functional connectivity analysis and use the ordering provided from structural connectivity, we can see there are strong similarities in the functional connectivity matrix that again gives us a sense of a first order validation that there's a lot of signal in both approaches. To carry this forward in the future, we will, as I alluded to before, need a very powerful informatics platform. And what we want to be able to is provide what we're calling a Connectome workbench that will link the user online to our emerging Connectome DB database that Dan Marcus has powering, and to allow queries such as, I'm interested in females with IQ of over 120 or with particular specific skills in, say, working memory, and give me that subset of individuals and let me look at the circuitry of those individuals and to compare them with any other group of interest. And this harkens to some of the query approaches that Rob was alluding to before that we hope to build forth on. So we have strong ambitions and our work cut out for us to take this collection of data that will be emerging through the Human Brain Project and making it a freely available and easy to explore and data mine for the community at large. But as I say, the informatics challenges are substantial enough that the efforts we can carry out in our own group will need to be supplemented by what the community of neuroinformaticians in the broader realm can add to the equation. So thank you for your attention.
Thank you very much. Very nice talk. Since we have little time, so I'd like to ask any question or comment on the floor. Okay. Wow, that's my response to this. This is going to be just outstanding. Will you be able to get morphometric data of volumetric data, just to basically do what we've been trying to do in mice, which is get modifiers for different chunks of brain?
Yes. So we will acquire high-resolution structural MRI, and we will carry out a set of analyses starting with the FreeSurfer software as it stands and as it may be evolved. So it does, for example, cortical thickness analyses on every individual subject. We have shape analysis tools, surface-based. There are subcortical parcellation capabilities already in FreeSurfer, and those are likely to become more advanced. So there will be the substrates for morphometric as well as connectivity analyses that can be put together synergistically.
Any other question? Yes, please.
Just a quick question. So will the 1,200 subjects include many ethnic groups?
So it will be, by the stipulation of NIH, capture the ethnic diversity of the United States as a whole. So we've got a strategy to represent.
So how many Asians do you think there will be?
That percentage, I forget whether it's 2%. It's whatever the known percentage is, and we plan to respect those important groupings. Yeah.
Okay. Thank you.
Okay. Thank you very much. Nice talk, David.